Hello, my name is Martina Summer-Kutmon, and I'm an assistant professor at Maastricht University, and I have a shared affiliation at the Maastricht Center for Systems Biology and the Department of Bioinformatics. And today I would like to thank the organizers for inviting me to present the WikiPathways project. As one of the WikiPathways architects, I would like to start with an acknowledgement page because there are so many people involved in a project that I will present today that it's good to start that way. Together with Alex Pico from the Gladstone Institute in San Francisco, we're leading a team of core developers and admin curators, but most of the content and the features from all the different tools come from contributors and pathway curators around the world. So this is a real community effort. I will start with a short introduction about WikiPathways and some of the associated tools before I'm going to present how WikiPathways is used in two specific projects that I'm leading at the moment. One is on pancreatic cancer and the other one is on COVID-19. So many of you might have heard of WikiPathways, but there's a couple of fundamental concepts that I want to highlight of our resource. So we can all agree that pathway diagrams are found everywhere and they're really useful. We see them in books, in papers, in presentations. They just are really intuitive for us to understand the biological mechanisms happening. So the utility to biologists as conceptual models is obvious. But if we would model them properly, they would become immensely useful for computational analysis and interpretation of the large-scale experimental data that we produce nowadays. So instead of making an image in a paper, we could create an actual machine-readable model that we can reuse and that other people can reuse as well. As an example, I looked at this example figure in a nice publication in Cell, where they describe the signaling pathway. The receptor is nicely visible in orange on the top, and then the whole signaling cascade. It's a very complex signaling pathway, and each element also has 3 different data types visualized. So they have visualization. These are the little arrows next to the data node boxes that indicate mRNA expression, protein abundances, and some even have data for phosphorylation status. So it's a very complex figure. If I would want to reuse this figure with a new data set, I would need to change each arrow manually, or I would need to create a new pathway figure if I want to study the same data set in a different setting. So if you would create this, and a colleague of mine did this, as a machine-readable pathway model, it's the same interaction, it's the same elements, the same proteins, but it's a machine-readable model that now allows you to take your data sets and create automatic visualizations that can be reused. They can also be displayed on other pathway models directly without additional effort or steps involved. So ideally, authors of pathway figures, so if you publish a paper in a journal where there's a pathway diagram, it's okay to have this static figure. But ideally, it would be complemented by a machine-readable model that can then be referenced and become directly part of the publication, which will increase reproducibility and reusability of the knowledge for other researchers as well. And this was also this is the reason why all these pathway databases started. WikiPathways is one of the commonly used pathway databases. It was launched in 2008 as an experiment in community-based curation of biological pathways. Already back then, and I'm sure we can all agree it's even worse nowadays, is that there's so many publications nowadays that it's really difficult to keep knowledge up to date, accessible, and integrated. So what we had the idea back then was, can we take advantage of the direct participation of experts, so of the researchers that actually study these processes, and this is called a concept of crowdsourcing. The most common website that works this way is Wikipedia. We all know about that. And WikiPathways is built on the same software as Wikipedia and focuses on collecting and curating knowledge. In our case, we focus on knowledge of biological pathways. And that also means that the database is community curated and everybody can contribute pathways, edit and curate existing pathways, and can use the pathway collections downstream. What are the advantages of such a system? This provides a platform where new findings can be added immediately, ideally by the experts themselves. If they find a new inhibition that plays a crucial role in the process, then this can be added directly to the model. Discussions about content are really encouraged, and there's a version history that allows easy reversion to a previous version if it's needed. We have successful collaborations with other manually curated pathway databases like Reactome and NetPath. It's really a complementary platform to the existing manually curated databases. Using the concept of quality tags, these are annotations which give information about the curation status of a pathway. We allow users to share and collaborate on pathways that are maybe still under development, or they might be hypothetical, so they will not be included in downloadable collections for analysis. And then using collections, ontology tags for diseases and cell types, we can allow users to upload pathways that are disease or cell type specific and annotate these accordingly, so other users immediately know what the pathways are about. This is a typical pathway page on WikiPathways. Every pathway has its own page where we have the title and the species, as well as the author list. The original author of the pathway diagram, so the one who submitted the original one, will always be on the first position. And afterwards, depending on the amount of contributions, there's sometimes a long list of contributors that increased the quality of the pathway as a whole. Below the author list, we have a clickable diagram. That means you can zoom in, you can click on the elements, and if you click on such an element, you actually get a lot of annotations and information about each individual element. That makes it not just an image, but it makes it a machine-readable format. So underlying each element, there's an identifier. In this case, it was annotated with an NCBI, an Enrichment identifier. But using BridgeDB in the background, we also get external references to a lot of other resources, including Ensemble, UniProt, and so on. So if you want to visualize your data and it's annotated with another database identifier than the pathway itself, this mapping is done automatically for you. Additionally, below the pathway, you can also find these quality tags. In this case, it's an approved version, which means it will be included in the collections that users can download. And they provide insights in the curation status and the biological context of the pathway model. Each element in the pathway, so the data nodes, but also the interactions, can be annotated with references to publications and other information. That allows you to create a nice bibliography of all the papers that have been used to curate and create this pathway model. As mentioned before, it's a wiki system, and that means that every single change in the pathway is recorded and stored and visible on the pathway page. And it's easy to check and compare the versions to see what has been changed and should it be reverted or not. It's easy to assess here. Every pathway page also has a discussion page on the top where we encourage people to really look and discuss the content of the pathway if they want to have additional elements or aspects in the pathway model. You can download the pathway in the bottom right with a drop-down box in different formats, from machine-readable formats for analysis to different types of image formats that you could include in your presentations or your publications. Additionally to the larger species-specific collections that I talked about for analysis, we also enable communities to curate and maintain their own collections focused on a topic of interest. We call these community portals. These are separate entry points to WikiPathways with an own front page where they can describe the project and the community. And we currently have over 15 different portals for communities like plants, rare diseases to COVID-19. Until now, over 700 contributors, researchers contributed to the knowledge base. They either uploaded a new pathway or curated at least one existing pathway. So it's a large group of people contributing to the content of our a little bit over 2,800 pathways. Our website is visited by thousands of people each month, users each month, And the programmatic access and the dissemination in other resources reaches a very wide audience and makes the use of the pathways very easy in your own research projects. In the last NAR paper, that is a database issue, we actually highlighted this focus of WikiPathways on connecting communities. So we focused on describing some of the communities that started portals on the platform, but we also highlighted the downstream tools and other databases where WikiPathways content is disseminated in. And that's the second part of my talk that I want to focus a little bit about. And that's the WikiPathways tools. The first one I want to mention here is our pathway editor called Pathwasio. It's a standalone application that allows you to draw your diagrams. So it's a visual tool. It works a little bit like PowerPoint, where you click and drag things over. But it also is creating a fully machine-readable model that allows you to annotate each of the elements, add publication references, add graphical elements, add annotations, and things like that. Pathwasio can also be extended with additional features, for example, for additional curation tests or visualization of data using the plugin system. And we also have a WikiPathways plugin that allows you to then directly upload your pathway to WikiPathways from within the application. There are several different ways on how WikiPathways data can also be accessed programmatically. We have a REST API, and our data is available in RDF format. So if you're using semantic web approaches to connect knowledge and data from different resources, you can also include WikiPathways within that. Additionally, we have an R package and a Cytoscape app. So if you use Cytoscape for network analysis, you can also use the pathways in that. And I have a little bit more information about that later on. Recently, we also put all our human pathways into Index. That's the network data exchange format, which is a very nice platform where all kinds of network resources are connected and easily accessible for users. And we also distribute the human content. Together with the Reactome team, we developed a bot to put our human pathways into Wikidata, which is a semantic version of Wikipedia, you could say. So for gene set enrichment analysis, which is often done when you analyze, for example, transcriptomics or proteomics data, we also have GMT files available, which are now included in MSigDB, which is the Broad Institute's collection of gene sets. I want to quickly highlight for the semantic web approaches our SPARQL endpoint, because that has been updated with a major interface update using Snorkel. That means that now users can start from a large set of example queries, which are shown on the right. There's simply a whole collection of queries that you can immediately run. Some include federated queries to Nextprot or queries about data curation. So if we have these pathway collections now, what can we actually do with these pathways? The most typical thing to do is perform enrichment analysis. So for example, you have two groups in your data set, healthy and a diseased, or different subtypes of a disease. You can check which biological processes, so which pathways are affected in the experiment. The outcome usually is a table like this, where you have the pathways and the different enrichment score shown. In this case, it's from a paper where we compared the frontal and temporal cortex of Rett syndrome patients compared to healthy controls. So we wanted to see which pathways are affected in which brain region, and if there's a difference between the brain regions. And we see that some pathways are affected in both brain regions, but some are uniquely affected within one brain region and not the other. If you have this table now, the nice thing that makes pathways very different from gene lists is that we actually have a visual diagram. But we have, this diagram is not just an image, but it's a model, which means we can visualize the data of this specific pathway that we know statistically is affected. We can actually visualize the data and look how the changes look like. So if we look at this example, it's the microglia pathway, where we visualize the frontal cortex and temporal cortex data next to each other. So each box is split in half. So for example, here the LAT, the LAT gene, is slightly downregulated in the frontal cortex and slightly upregulated with a positive fold change, a log fold change. It's upregulated in the temporal cortex. And we get an immediate overview of what is actually going on in the pathway. And we can now see how Rett syndrome patients, how the pathway in Rett syndrome patients is affected and regulated differently than in healthy controls. Lastly, I also want to highlight the usability of these pathway models. So they're not just nice to create a visual representation, but we can use them also in more systemic analysis, more global analysis, for example, in network analysis. So we could extend the pathway with regulatory information or drug target information, which is often excluded when we draw the pathways because we don't want them to be really cluttered and we want to make them very easily readable. But they can be easily added with one click. You can add all of these elements. Additionally, it's useful to study, for example, the overlap between pathways. We often assume these pathways are completely independent. And if we look at the image, we don't see the connections. But actually, these pathways are not independent, and there's a lot of overlap, and maybe they share even sub-pathways, and that can be visualized in networks as well. And I will show that in the use case a little bit later as well. From a network perspective, we have developed the Cytoscape app for accessing and importing pathways in Cytoscape directly. And we have two different views. So one is the traditional pathway view on the left, which is how it would look also like in PathVisio or if you would download an image. And on the right, it's the network view. So it's the same pathway, but the layout is gone, all the graphical elements are gone, and we can now actually perform data integration, network extension, and network analysis with this version of the pathway. I'm going to show how some of these tools are actually used in practice. And I'm going to do this with two projects. So the first project is a pancreatic cancer project, which is funded by SonMV and DTL. And the aim of this project is to create a knowledge resource for EU pancreatic cancer translational research projects, where we on one hand want to create a curated set of pathway knowledge. So we want to have kind of a pathway collection, a pathway portal for pancreatic cancer, where we have general cancer pathways that are relevant, but also specific pathways for pancreatic cancer. And then on the other hand, we want to provide automated analysis workflows. Most of these are developed in R and using Jupyter notebooks, where we provide molecular data analysis workflows with a focus on RNA sequencing data and nanostring immune panel data. And the aim here is not the pre-processing of the data, but it's actually really focused on the interpretation, so the process level analysis and the visualization of the data in the biological context. So in one of these workflows, we used a subtype data set of pancreatic cancer. So there was a study in 2015 where they used 170 pancreatic cancer patients, and they identified a signature of two clear distinguished subtypes, and they call them the classical and basal-like pancreatic cancers. I'm sure most of you are aware pancreatic cancers is one of the cancers with a very bad prognosis in generally, as they're normally diagnosed at a very late stage where they often already have metastasized. So better understanding and maybe improving the prognosis in a sense is really important for this cancer. So they nicely showed that this signature separates the classical and basal-like subtypes very clearly, and the classical has a much better prognosis than the basal-like patients. So what does this workflow do? It's completely automated, and it can generate these figures. It generates each of these figures and additional ones. So we first compare the two different pancreatic cancer subtypes, looking at differential gene expression. So we want to see which genes are up and down regulated, and do they share them? And we see a lot of genes are actually shared. For example, 515 genes are upregulated in both the basal and the classical subtype, but there's a lot of additional genes that are upregulated only in the basal or only in the classical subtype. There's only 3 genes, one for the downregulated classical down and basal up, and 2 for the classical up, basal down, that actually switch their expression changes in the between the subtypes. So while in the classical it's downregulated compared to the healthy control, in the basal one it's upregulated. So those are interesting genes to look at as well. Next, we perform these pathway enrichment analysis that I also described before. In this case, we use an R package, and we can create figures like this, where we then see how many genes are present in these pathways and which ones are significant. If we look at these pathways, a lot of them are immune related: focal adhesion, interleukin 18, wound healing, and so on. And we can now start to actually look at the diagrams, because we don't stop here with the list of pathways, but we can look at the pathways. And so this is an example of a pathway where we can actually look at this pathway. Again, we split the boxes into half. Just checking if I can use the laser pointer here. We split the boxes into half. On the left side, we see the basal subtype. On the right side, we see the classical subtype, and we see a much stronger response in the basal subtype in this pathway compared to the classical. This is just a small snapshot of the large pathway. And that might lead actually to this more extreme, more worse outcome for the basal subtypes. So that might indeed lead to certain insights on the differences between these subtypes, and how the basal subtypes might be needed to be treated differently than the classical subtype. And the last part that I want to shortly mention here is this overlap analysis, this crosstalk analysis. So we can use all the pathways that are changed and see which ones are affected. For example, here the vitamin D receptor is only present in the vitamin D receptor pathway from the changed pathways, but a lot of the genes are also shared. The second project that I want to highlight quickly is the COVID-19 project. The COVID-19, well, we're all very strongly affected by it, obviously, and already in March 2020, we as a community realized very quickly we need to create pathway models to better understand what's happening, and we started this portal. Obviously, we didn't do this alone, and actually the pandemic brought together a community of over 200 researchers from around the world to collaborate in the disease map project, COVID-19 disease map project, which just this week, the paper in Molecular Systems Biology was published. So we tried to give an overview of the complete molecular mechanisms, from the virus attachment to the entry of the virus in the cells, to the virus replication, to downstream effects, and so on. If you click on one of these pathway models, you get again this visual representation of the pathway model, and you can use it for data analysis, visualization, modeling, and so on. We're still working on this. We still have weekly talcons to streamline the curation efforts between the groups, but it's a very broad community with biologists, clinicians, data producers, text mining, computational modeling, and data analysis, and so on. So it's a really fun project, actually. In this project, I also got a grant, and we really wanted to focus on this continuous reproducible data analysis, because there's so much new information coming all the time. Every day there's something new that we need to test. And this is one that I will quickly show how we use WikiPathways and the disease map pathways for the analysis. This is just a data set that we used as an example. It's a human lung epithelial cells that were exposed to SARS-CoV-2 or a mock virus. We do the enrichment analysis. We see a lot of COVID-related pathways. All the ones in purple here are the ones that have been curated specifically for COVID-19. And we can visualize the data and see how this interleukin, this type 1 interferon pathway, this actually is strongly upregulated in the COVID-exposed cells. So again, positive fold change means more expression in the COVID cells, means a red color. Downregulation would be a blue color, but there's very few. There's just 1 or 2 that have a slightly blue color. There's also a possibility to create this pathway gene crosstalk again. So we see these pathways have a lot of overlap because there's always immune components and things in there. But it's important to see where the pathways overlap and how they overlap. We also extended these with drug target information from a drug bank, for example. And so there were 444 known drugs, and a lot of the actual currently experimentally unapproved treatments of COVID-19 are included. For example, tocilizumab, an interleukin-6 inhibitor, which is shown up here, is one of these that we also immediately found looking at the analysis that we performed. The last part that I wanted to show, let's see if I can move to the next page. Turning off the laser didn't work. Let's see. Now it should work. Yes. There's a lot of information still missing, right? Because we're curating all the time. Currently in pathway databases, we have about 60% of protein coding genes that are included in the pathway models. So we need to increase that percentage. And we wanted to do the curation a little bit more targeted. So can we see what's actually affected in the cells, in the humans, and so on, in all the data sets that we analyzed, and therefore prioritize certain proteins and certain pathways to be looked at. So in this case, we can see these are all the differentially expressed genes and their functional relationships using protein-protein interactions. And we can see that there's a whole cluster here that's strongly upregulated, but it's not present in any of the COVID pathways yet. So it really gives us hints to where we should go with the curation efforts, let's say it like this. So we can really see what are we still missing in our resource. Very recently, we also started to look at pathway activity in different tissues. So we actually use protein expression data from a lot of different tissues, 7 in total from COVID patients. This is really work in progress, but we can nicely see how certain proteins are only expressed in one tissue or in all tissues, depending on the type of protein we're talking about. And we can compare that with the protein expression in healthy subjects. So in summary, I hope I was able to show that WikiPathways is a community-curated pathway database. Maybe you're now kind of encouraged to start curating some pathways yourself that you might be working on. We are very welcoming to communities that want to use WikiPathways to create pathway portals to make their own collections of pathways that are relevant for a specific biological topic. And for my personal interest, I think the importance of this automatic analysis workflows is essential for making analysis reproducible, open, other people can reuse it for their own data and so on. And that's essential for studying disease mechanisms when we want to identify affected processes, see how pathways are interacting with each other, or maybe we want to investigate known drug target information. These are just some of the examples that you can do. There's a lot of people involved in the different projects. In WikiPathways, obviously the developer and admin curation team, but also all the international pathway curators and developers that develop all the tools. All the tools are actually open source. They're all on GitHub. We're always welcoming to new people that want to contribute new features. Also the PanCANnet project, the Pancreatic Cancer Project with Erasmus Rotterdam MC and Omnigen, and the COVID-19 project, which is funded by SonMW, and obviously also the whole COVID-19 disease map community led by Marek and Anna. And I'm happy if there's still time to answer some of your questions.
Yeah, wonderful. Martina, this was actually really interesting. And we've got 2 questions in the chat. And I've got a question that I really want to ask. But let's give priority to our conference attendees. So The first question is from Anugera Herlaut, and it's about the data representation used by WikiPathway and whether it allows for computation. So he says it'll be quite interesting to be able to combine and analyze different pathways. What are your thoughts here?
Yeah, so I need to be careful with computation. I assume it's meant that you model it, so modeling in the sense of simulating data. Some pathways in WikiPathways, so within the COVID-19 project, we actually can convert all our pathways to SBML or SPGN, so there's definitely possibilities. Since it's a very open community, it means that some communities choose a specific drawing style that might not be ideal for this kind of modeling approaches, but many of the pathways can definitely be used. And we integrate and combine different pathways very often. Although the combination within the nice layout, let's say, is a little bit harder than if you look at it from a network perspective. So I hope that answers the question.
Yeah, no, that's really interesting. Question from me was really just about the kind of commercialization aspect of this, a potential commercialization aspect of this. So when you were talking about WikiPathways, I got flashbacks of using this program called Ingenuity Pathway Analysis, which I thought was quite similar. And actually, I learned recently that IPA sold to Chiagan for, I think, as much as $100 million or something like this. So there's definitely a lot of value in ontology and ontology, and WikiPathways is just an open source effort. We're getting open source companies springing up left and right. Elasticsearch is a great example. MongoDB. What are your thoughts here? Yeah,
so at the moment we're really funded by by grant monies. So we really research communities, let's say it like this. The development is really currently funded part of that, and I think that's also our whole mindset a little bit. So we are very open. We want to collaborate with others, and for now it works very well. So I don't think we will change this very quickly. Also because the content that's provided is really coming from the researchers themselves, right? So we wouldn't own any of that information anyway. We just really want to collaborate with all the people involved and help researchers to get as much out of their data as quickly as possible, I guess. So I don't think that we are gonna go into a kind of company style or anything like this.
Well, if you change your mind, let me know, and then,
I will.
and then we can work something out. Okay, yeah. Wonderful. Martina, this was great. We do have to move on to the next speaker in the interest of time.
I will try to answer some of the questions in the chat.
There were still questions in the chat, so if you could deal with them offline, I'd really appreciate that. And thank you so much for being here.